Hello and welcome to our presentation on DNA replication. DNA replication is the biological process of producing two identical replicas of DNA from one original DNA molecule. DNA replication takes place in the nucleus of eukaryotic cells and in the cytoplasm of prokaryotic cells, because as we know, prokaryotic cells contain no nucleus. So, DNA replication, why is it important? If DNA never replicated, meiosis or mitosis would halve the size of the genome until the cell would die, and that wouldn't take a lot of time. I'm talking about DNA replication, but what exactly is DNA? DNA stands for deoxyribonucleic acid. DNA contains nucleotides, and these nucleotides consist of a phosphate group, a sugar group, and nitrogen bases.
Initiation. The purpose of initiation is to focus on binding DNA A initiator protein to the DNA, so it begins initiation. It is the pre-replication process. It also involves unwinding the DNA to proceed to produce a replication fork. What proteins are involved during initiation? There are a number of different proteins involved. These include DNA A and the origin of replication. These function as a protein that activates the initiation of DNA replication to the origin of replication sequence to activate replication. So the DNA A attach onto the DNA double strand to initiate replication at the origin of replication. CDC6 and CDT1 protein complexes both function the same. They help MCM protein complexes move into the double strand of DNA. MCM, mini-chromosome maintenance protein complex. This is a DNA helicase. It is a class of enzymes, which is a motor protein. It has 6 gene products, MCM 2 to 7, form a heterohexamer. GINs and CDC45 proteins both have the same function. They both unwind the DNA, as shown in the structure there. So the process of initiation. DNA A move on to the double helix DNA strand at the origin of replication. MCM, helped by CDT1 protein complexes, move in to the origin of replication. The CDT1 move out once the MCM protein complexes have moved in. The GINs and a CDT45 help also MCM to unwind the DNA to form replication fork.
So in this part we are going to talk about elongation. Elongation is the second phase of the DNA replication process. The rate of elongation is about 500 nucleotides per second in bacteria, where in eukaryotic cells it's 50 per second. DNA polymerases are present in eukaryotic more than prokaryotic cells. So as the structure of DNA strands are different, means that they are arranged in opposite sides or antiparallel, DNA polymerases, which are important enzymes in DNA replication, are forced to add nucleotides to one end only, which is the free 3 end. So as a result, the new DNA strand will elongate in the direction of 5 to 3. Leading strand is one of 2 mechanisms that occur during elongation. In leading strand mechanism, after RNA primer is made, DNA polymerase 3 is in one template strand can synthesize a new complementary strand by elongate the new DNA strand in the direction of 5 to 3. DNA polymerase remains in the same replication fork with that template strand, and with the help of protein called sliding clamp, it can move along the template and keep adding nucleotides as the fork progresses.
I will introduce the elongation, the process of the DNA replication. As well known, DNA has two double helix strands. One of them is the leading strand. The 3' end to 5' end preceding doctor strand that uses a 5' end to 3' end template is called leading strand. And the lagging strand, the replication of this template is complicated and the new strand is called lagging strand. Synthesis of the lagging strand persists discontinuously, initially forming the Okazaki fragments from multiple RNA primers that are formed on the parental strand as each new region of DNA by DNA polymerase. The RNA primers are necessary for DNA polymerase to bind nucleotides to the 3' end of them. The doctor strand is elongated with the binding of more DNA nucleotides. As each growing fragment approaches the previous primer, that primer is removed by another enzyme, and the fragments are joined by DNA ligase to form a continuous DNA strand. By repetition of this process, the entire lagging strand eventually is completed. The gaps are closed with the action of DNA polymerase and DNA ligase. DNA polymerase adds complementary nucleotides to the gaps, and DNA ligase adds phosphate in the remaining gaps. So now we had a question. Why is DNA synthesis continuous on one template strand and discontinuous on the other template strand? I think it's because the DNA synthesis proceeds in the 5' end to 3' end direction only, yet both strands of DNA must be replicated and are oriented anti-parallel to one another. Therefore, one strand must be made in short pieces called Okazaki fragments to accommodate overall 3' end to 5' end strand growth, while maintaining DNA synthesis in the 5' end to 3' end direction. In the lagging strand, the DNA polymerase I exonuclease reads the fragments and removes the RNA primers. This picture shows the whole process of the elongation. The first one is RNA primer copied from DNA, and then DNA polymerase tree elongates RNA primers with new DNA. And DNA polymerase I removes 5' and RNA at end of neighboring fragment and fills gaps. The last step is DNA ligase joins adjacent fragments.
Now we come to the final step in DNA replication called termination. The two main enzymes involved in termination are DNA polymerase and DNA ligase. After the DNA polymerase has done its job, the newly synthesized DNA looks something like this. It has short segments of DNA with interrupting sequences of RNA primer. After this, another enzyme known as the exonuclease comes into play, which removes the short segments of primer. These are then replaced by short new segments of DNA by the DNA polymerase. After this, the enzyme DNA ligase joins or seals the DNA fragments to produce a continuous strand. Lastly, it is also very important to check for errors in the newly synthesized DNA, and this is done by another type of DNA polymerase which proofreads the newly synthesized DNA for any wrong bases that may have been inserted, and if it finds any wrong base or sequence, it removes the base and replaces it with the correct one.
After the process of DNA replication is completed, the two newly synthesized DNA molecules coil up into the DNA helix model. In this way, the semi-conservative nature of DNA replication happens. And with this, we come to the end of our teaching presentation. Thank you.